Hello guys, welcome back to the another concept in human nervous system. This time, nervous system is continued with the sense organs. We have five important sense organs, as you know: eye, ear, nose, tongue, and skin. But in the syllabus of NCRT, for NEET orientation or any pre-medical examination, we have to go through only two important sense organs. One is eye, and another one is ear. This class I am going to explain about eyes. Eyes are present many animals. In the early of the animal kingdom, eyes were absent. Simple eyes, compound eyes, false eyes. Whereas come to the vertebra, in fishes and amphibians, reptiles, apes, eyes with nictitating membrane, where that nictitating membrane protects eye from the different mediums where they are living. In case of mammals, that nictitating membrane is reduced, becomes vestigial. In the corners of the eye, you can observe as a little bright colored bulb-like structures. That is third eyelid called nictitating membrane. It is functionless. We have two eyelids, upper eyelid and lower eyelid, whereas upper eyelid is movable, lower eyelid is not movable. But in the reptiles and fishes, birds, if you see, eyelids are different. They are moving from sides and up and also from upwards, downwards and upwards. We have eyelid movement only downwards, like when we are blinking eyes, they are only moving. And eyebrows are present only in mammals. They are protecting eyes from any of, suppose, sweat, during, in case of bathing, during in case of rain. So they are just act as a shades. Eyebrows, eyelashes are protective structure for the eye. They are associated with the human eye structure just because of a mammal. Internal structure, anatomy, if you see the eye, human eye. One pair of eyes are present. Let me give you a complete structure of human eye anatomy first. So, eye internal structure have three regions. Upper, outermost region. This is posterior part. This is anterior part. Posterior part is called sclera. Sclera. Generally, sclera is white portion of the eye. White portion of the eye. In the eye center, you can see the dark structure, and around it there is a white portion that is simply called sclera. What is sclera? It occupies first layer, anterior side, posterior side. This is anterior side. Where in the anterior side, this part is called cornea. Cornea. Cornea and sclera both are occupying first circle, first order. But this is posterior side. This is posterior side. This is anterior side. Anterior side. First oral of the eye occupied by two structures, sclera and cornea. Cornea is anterior part and the sclera is posterior part. Further, cornea gives another protective layer or conjunctiva. This is conjunctiva. This entire, all this is the first layer of the second layer. Below the sclera, there is a choroid. One side I have given shade, another side plain to understand. This is the first layer, this is second layer. This is called choroid. Choroid is second layer. As I said, sclera is the white portion of the eye and choroid is blue portion of the eye. Blue portion of the eye. Don't confuse. I didn't say that eye is blue color. In every one eye, whether your eye color is brown or black or different shades of color, in every person, choroid is blue in color, but the blue is below the white. As white is dominating the blue, in that we cannot observe the blue. But choroid is blue. Choroid is present which side? Posterior side. Which layer? Second layer. So, sclera is first layer. Cornea is first layer. Choroid is second layer. Below the sclera. Posterior, anterior, posterior. Anterior side, the choroid becomes ciliary muscles. Becomes ciliary muscles. So ciliary muscles are extension of the ciliary muscles. Ciliary muscles are extension of choroid anterior side. This is second part. This is also second part. All the parts are towards anterior side. All these parts are towards posterior side. I hope you are understanding, right? No confusion. This ciliary muscle, the second oral, is not yet finished. But it will not become circle, no? The ciliary muscles are producing some extended portion is called iris. Iris. Iris is called color portion of the eye. Color portion of the eye is iris. If the iris is blue color, your eye look like blue color. If iris is brown, your eyes can be judged as brown eyes. So color of the iris accomplishes the color of the eye. So white portion of the eye is sclera, blue portion of the eye choroid, color portion of the eye. Who decides the eye color is iris. Iris is the extension of ciliary muscles. Then here you can also see lens. This is called lens. Lens is also occupied in second oral. Iris is also occupied in second oral. Ciliary muscles are also occupied in second oral. First oral composed of sclera, posterior side, cornea, anterior side. Whereas second oral, posterior choroid, same choroid, anteriorly finished as ciliary muscles, lens, and iris. Iris is incomplete part here. You can see this is what? Iris. And this opening, the gap here, it is called pupil. Pupil. This place helps to contract and relax by the help of ciliary muscles during watching of an object from far and also near. If you want to see an object from far, which is very far, you have to see from here, then you have to contract your pupil so that objects can be very clear and visible. If it is the blur also, you will use to contract the eyeballs to see them very clear. Whereas the nearest objects, we have to expand our eyeballs. So what is this? This is simply usage of eye to observe the objects near and far. Then choroid, ciliary muscles, iris, and lens, everything. Pupil is not a part, which is the region between the upper and lower iris part, which is just the opening part like structure. And last one, that's the third one, is called retina. Retina. So retina is the innermost coral of the eye anatomy, and it consists of again 3 structures. So this is retina. This is retina. Retina is innermost layer. Innermost layer. Retina helps to visualize the pictures. Let us see how that is the physiology, mechanism of vision. I will explain after this. One should understand anatomy, structure of eye, what are the parts present in the eye structure, where exactly they are located, and what functions of everything is next part. And now if you see this cavity, this is called vitreous chamber. Vitreous chamber. In the vitreous chamber, vitreous fluid is present. It is crystalline nature. Once you touch the cell phone screen or a plasma TV, LED TV and all, if you give a little pressure, then you feel that some fluid is there inside the screen, beyond the screen. Just like that, beyond all these layers, some fluid will be there called vitreous fluid. We can define that vitreous fluid is present between lens and retina. Lens and retina, this place is called vitreous chamber. Vitreous chamber is filled by a fluid is called vitreous fluid. What does vitreous fluid do? Vitreous fluid amount is sometimes less, then eyeball size is less. Vitreous fluid amount is large, then eyeball size is large. Suppose if you see Chinese people, their eyeballs are very little in size. Means vitreous fluid is very less in them. Genes are maybe the responsible. A protein may be a gene responsible to secrete the vitreous fluid is already adopted for that. Maybe that is the reason one we can say Chinese eyes are very little and narrow eyeballs. Whereas in other country, even in India also, in some people eyeballs are very large and protruded structures. This is not because of any kind of a thing except the amount of fluid. Amount of fluid is rich, eyeball size is rich. Amount of vitreous fluid is less, eyeball size is less. Normal, then normal. So size of the eyeball is decided by the amount of vitreous fluid. That's it. Where does this vitreous fluid is present? In vitreous chamber. Vitreous chamber is located between the lens and retina. Got it? Now, then between the lens and cornea, this is cornea. Between the lens and cornea, this place is vitreous chamber. This place is called aqueous chamber. Aqueous chamber. In the aqueous chamber, aqueous fluid is present. Aqueous fluid is present here that helps in the diffraction of light. But this is decides the size of light. These are important structures. Other than that, if we talk, here blind spot is present where imaging is not formed because of absence of photoreceptor cells. That is also part of retina. From the blind spot, optic nerve will be leading. Optic nerve. Yesterday class, if you remember, optic chiasma present in the midbrain, aqueous duct, and optic nerve becomes a cross-like structure. This is optic nerve. From one eyeball, optic nerve will go, another eyeball, optic nerve will go, both together jointed and go through the midbrain, and the final destination for them is optic lobe of the forebrain, not occipital lobe. So there they will go. But what I am saying here, they are leaving from the blind spot. From the blind spot area, they are leaving. This is optic nerve. Our direction of optic nerve is towards optic lobe in occipital region. Occipital lobe of forebrain. Understand? Good. Now, in the retina, a certain area which is called fovea, it is also called yellow spot. This is blind spot. This part I didn't clearly here. Sorry. This is blind spot. Macula densa. This is a fovea called yellow spot, where only cones are present. Where only cones are represented. Brightness and high frequent light pictures can be captured in the fovea. This is the anatomy of the eye. Let me revise it in 1 minute. Human eye structure consists of 3 layers: outer sclera and middle choroid and inner retina. Sclera, which occupies only posterior part, the same sclera anteriorly become cornea and conjunctiva. And in the second layer, choroid posteriorly exists, and the choroid modified into ciliary muscles, lens and iris in the anterior part, all together in the second wall. Third wall is completely occupied by the retina. Retina is made up of photoreceptor cells. That photoreceptor cells are absent here. That is called blind spot. At the blind spot, from the blind spot, optic nerve leads towards the optic lobe in the forebrain. And the fovea is the region where photoreceptor cells are rich, mainly cones. They form very bright pictures. Clear? And this is the vitreous fluid present in the vitreous chamber. This is aqueous fluid present in the aqueous chamber. The lens is also part of ciliary muscles extension, which used to capture the picture. And the movement of lens is decided by the ciliary muscles, in and out, zoom in and zoom out to capture the pictures near and far. And the extension of ciliary muscles towards the conjunctiva is called iris. Actually, the color of the eye means iris color. Then between the iris, there is an opening like structure. For our understanding, I have given such a big gap, but actually that is not a big gap, very small gap. That is called pupil, which helps the contraction of eye in the observation of objects. This is anatomy of eye only in the purpose of NEET. If you are an ophthalmologist, eye specialist, more than this structure you have to be learned. For the purpose of attempting the NEET examination, this is enough. Now you are going to learn about mechanism of vision. How eye exactly works to provide vision for us. Concept eye is mechanism of vision. Mechanism of vision. Come, let's know how we can capture objects in front of us in only light. This is the eye, where this is one object, maybe a person. Between the person and eye, there should be light to perform the mechanism of vision. The light, definitely, there is no doubt, sunlight. In the sunlight, there are so many rays of there: alpha rays, gamma rays, cosmic rays, infrared rays, visible rays, and ultraviolet rays. Many rays are there. Their frequencies are different one from another. Then, which frequency is exactly required to capture an object? That is 400 to 700 nm, which is present in visible light. Visible light frequency is 100 nm to 1000 nm, whereas 400 to 700 is present in between the 100 and 1000. That is why. Lesser than the 400 and greater than the 700, human eye cannot capture. Another animal eye can be captured. Dog can see, owl can see, ox can see, different animals, birds can see, rather than this and greater than this. But we can capture only. It means beyond the 400, greater than 700, there will be a world, but we cannot see. What we see is our world. What we cannot see is mystery. So that physics can explain what actually present out of this both the frequencies. Okay. Now let us talk. Don't drag me into the physics. You and I don't know. Then when a picture picture in the sufficient light is captured, then by the iris it is captured and it hits the retina. It hits the retina. First it comes to conjunctiva, later aqueous fluid, the photon, that aqueous fluid to lens, lens to vitreous fluid to, it hits the wall of retina. Why? Because it is the innermost wall, innermost layer of the eye, so that it has to be hit the wall of retina. You should observe that retina wall. Retina wall. Suppose entire this is retina. I am explaining this part. This part. So this curve is this curve is this. I am taking a piece of egg or pizza. So this is innermost part of the retina wall. This is outermost part of the retina wall. It is composed of 3 layers. Don't think that I am again exploring the wall of eye. Wall of eye have 3 layers: outer sclera, middle choroid, inner retina. Inner retina again have 3 layers. That is what I am explaining. This is innermost wall, this is middle, this is outer. Inner wall of retina, middle layer of, layer is better, not wall. Inner layer, this is a middle layer of retina, and outer layer of retina. Outer layer of retina is closer to choroid. Inner layer of retina is closer to vitreous chamber. Let us see in the inner layer, so many ganglion cells are present. These are ganglionic layer, we can call them. This is ganglionic layer of the retina. We can call this is ganglionic layer, innermost. Whereas in the middle, bipolar neurons are present. Bipolar neurons. I already told you the examples at the time of neuron types. That is bipolar neurons. They have a 2 poles. One pole is in the side of ganglions and another one is in the side of photoreceptors. Photoreceptor cells are present in the innermost. Okay. Then this is innermost wall, middle wall, middle layer. Then it is connected to the photoreceptor cells. Photoreceptor cells composed of rods and cones. Like this. These are cones and it will be rod. Rods and cones together called photoreceptor cells. Let me write. The middle layer is composed of bipolar neurons. Bipolar neurons, and innermost layer, so outermost layer is composed of photoreceptor cells. Photoreceptor cells are 2 types: rods and cones. Rods and cones. If you see the rods, they have a specific protein which in the composition is rhodopsin. Rhodopsin is a protein present in the rods, whereas in the cones, iodopsin. The ratio between rods and cones is almost 6,000:1. More rods and less cones. This is inner wall, inner structure, internal structure of the retina. This structure, if you know, then you can easily understand mechanism of vision. See, as I explained just before, a picture can be captured in the visible light, that is a 400 to 700 nm. Then it will convert it into photon. The photons are captured by the lens. They are passes through the vitreous fluid and hits the retina. If this is the same one, when it hits the retina, actually it will become reverse. How it will be reverse? I will explain. When the photon hits innermost part of the retina. Then ganglion cells are transferred that impulse to bipolar neurons. Bipolar neurons, the end which is representing the size of ganglion cells are not generating any kind of action potential. Action potential can be generated at photoreceptor cells here. Here action potential of the photon can be placed. Suppose first rods. If you see, this is the rod. This is what? Rod cell. Rhodopsin. Rhodopsin. Rhodopsin protein is present in the rod. The picture which captured to the rod, not directly to the iris. Lens to vitreous fluid to wall of the retina. Wall of the retina, first part is ganglia to bipolar neurons to photoreceptor cells, rods. When it hits the rods, then rod consists of two things. Rhodopsin consists of two things. Rod and opsin. Here opsin is a protein. This proton directly hits one part. Then this part will be flipped. It will become ulta. So what happened here? Photon plus opsin comes together out. Invertly reverse. Explain. If this is rod, then photon is coming in this way and hits. Actually, it is a rod. Rod consists of rhodopsin. Rod cells consist of rhodopsin protein in the opsin protein, and this is the light. Light means photon. Light plus opsin is coming this side. The flip. If it is like this, when it hits the light, then it will be flipped. The path is invert. So that invert image will form at rods, means retina. Invert images are formed in which layer of the eye? Retina, because of the rhodopsin. Now, photon plus opsin is going towards the cones. Actually, cones are available in three colors: red, green, and blue. RGB. Not all different colors are present in our eye. Only red, green, blue. I will give you a best example here. When we give a print from system or phone to the Xerox machine, then a colorful print will be. But actually, there are no different colors. Not even 7 colors of the rainbow is present in the Xerox machine. Only 3 colors. They are red, green, blue. But how the different colors are coming, different shades are coming, mixing of colors are coming, means there is a frequency. One object which is coming chill with here as a photon that has a frequency, it will grab frequency of red, some percentage, and greater percentage of green, and less percentage of blue. Combination of these three provide different colors. Suppose if you have a red color paint, green color paint, and blue color paint, if you take 1 drop of red, 2 drops of blue, 3 drops of green, you mix, there is a different color. You change the ratio, 3 drops of red and 1 drop of green and 2 drops of blue, different color. If it is 10 drops, 1 drop, it is a different color. So many shades will be produced by the combination of these 3 colors. Now, what is my concept? Here, picture is formed, but this is black and white picture. Black and white picture. Black and white pictures are formed because of only rods, not by cones. Where does these cones, the rhodopsin, protein, photon plus opsin is coming to the cones, by the combination of the 3 color images are formed. Color images were developed in this by iodopsin protein. Thing is, from here to here, there will be the role of vitamin A. Vitamin A. What is the vitamin A name? Keratin. So, keratin protein is required to form pictures in the visual organism. That is why that is present in A vitamin. Deficiency of A vitamin Okay, xerophthalmia, night blindness, some genetic disorders also there. Okay, that is a different part. Vitamin A is very important in the mechanism of vision. Clear? So rods can provide only black and white images. By the help of cones, only color images, original color images will be formed. Suppose if it is yellow color object, here yellow color object will become. But actually the original yellow color is not there because yellow color cones are not there. Green color cones are there, blue color cones are there, red color cones are there. Some frequency of all the three becomes yellow color. That is decided by the photon. That's why photon is still carrying. I repeat, mechanism of vision. Last time, when picture was captured in the available light, that is 400 to 700 nm between, that is also required for photosynthesis, same frequency. Then, by the lens it is captured and it comes to the vitreous fluid. Vitreous fluid to hit the wall of retina. In the retina, first layer is ganglion cells. They receive the photon. Their image is not formed, and the ganglion cells sends the photon to bipolar neurons. That time also image is not formed. Bipolar neurons are sending the photon to photoreceptor cells. There, between the bipolar neurons and photoreceptor cells, here action potential can be generated. Action potential can be generated. When action potential can be generated, black and white pictures only can be formed. Black and white pictures only can be formed. Reason: rhodopsin invert the picture by the help of opsin and projected photon. Sorry, photon, not proton. Then the photon and opsin together gives invert image that is available in only black and white. If you see a black and white image, mechanism of vision is over. You capture a color image, then it will be continued. Then this inverted black and white picture goes to cones. By the frequency of gathering of the three colors of cones, your original color image will be formed. So frequencies are there in our eye. That is why different colors we can see. Color blindness means what? If the cones ratios are less in your eye, we cannot see the color objects. Suppose some person cannot see the red color, protonopia disorder, maybe genetic disorder. Some persons cannot see the blue color, okay, leukonopia. Some persons cannot see the green color, deuteranopia. These are extra part of this monoculism. But color blindness is different, night blindness is different. Regarding eyes, the disorders are also xerophthalmia, eye dryness, that happens because of the less percentage of A vitamin in our body. A vitamin helps for good eyesight. Okay, this is what the class about human eye anatomy and also mechanism of vision. I hope you have worked your eyes in the class. Thank you. I'll get you the next class with ear structure and mechanism of hearing. Thank you. Bye.